add to our network, and we hope it will be valuable for you.
Yes, thank you so much, Dr. Smith, for the invitation. I've had a great visit so far, and I'm looking forward to telling you more about how is it that I got to be where I am today in terms of being a scientist at a liberal arts university in the South. And so I was instructed to tell you a little bit about everything, about my science, how I evolved as a scientist, how my interests changed, but also I was recommended to tell you about how is it that I ultimately chose this path as an outlet for my science and for my work. And so you'll see that throughout the talk I'll be talking a little bit about science, but also about my professional development in general. And so I hope that will be useful to you. When I think of where I am as a scientist today, I think of myself as a cell biologist educator. And so an alternative title for my talk would be, if I had to pick one, would be, becoming a cell biologist educator. And it's been very gradual, very organic. And so hopefully the story that I will be telling you today will maybe ring true to some of your experiences, and hopefully you'll be able to maybe acquire some ideas, tips for success. As scientists, we all share characteristics, and I think these are some of them. We have curiosity, we are to some extent creative, and we are persistent. To the extent that we display these characteristics, that almost helps us shape our scientific careers. And that has been very much the case in my career. And my journey as a cell biologist has been defined by my curiosity for the natural world. Okay? So I'll tell you a little bit more about my academic training and how is it that I came to be a cell biologist today. As an undergraduate, I attended University of Miami, go Hurricanes, and I double majored. Oh, sorry. Excuse the plug. I double majored in biochemistry, molecular biology, and chemistry. So early on, I had this passion for math and biology combined together. I experienced research as an undergraduate, not only in the context of the research lab by doing volunteer work at research labs, but also through lab courses. I remember taking this transmission electron microscopy course that really changed the way that I thought about biology and about bench work. This idea that you could take a technique and use it to your advantage and could tweak it protocol-wise to give you what you needed was very fascinating to me. And then towards the end of my career as a senior, believe it or not, I took this awesome physical chemistry class. And it wasn't as hard as I thought it would be, and I really enjoyed it. And my passion for biochemistry, physical chemistry, and biology ultimately motivated me to apply to a graduate program in molecular biophysics. And so ultimately, I chose to attend graduate school at Yale in MBMB, what they call it for its acronym, and it was a great experience. When I started out as a graduate student at Yale, I found transmembrane protein super interesting. This idea that transmembrane proteins sit at the surface of the cell and are the connection between the outside and the inside of the cell. They can do a variety of functions. They connect the environment to the inside by being able to relay messages chemically. I found the idea of their structure fascinating, this idea that membrane proteins have to accommodate not only their function, but also the idea that they live in both a hydrophilic and hydrophobic environment. So I found them fascinating. I liked that challenge of studying membrane proteins. And so when it was time to join a lab, I ultimately chose a lab that would allow me to gain expertise in studying membrane proteins. As a graduate student, I chose to study a protein called BCX1. BCX1 stands for vacuolar calcium exchanger 1, and it's an intracellular transmembrane protein that sits in the vacuole of the budding yeast. So it's a protein from budding yeast. What its function is essentially it sits at the membrane of the vacuole, and it helps sequester calcium ions while exchanging protons. It recruits, or it sequesters those calcium ions into the inside of the vacuole. And in doing so, it helps keep homeostasis with regards to calcium in the cytoplasm. My work, very little was known about BCX1 in terms of its function and how it was carried out, how its structure accommodated its function. And so my job as a graduate student was to do structure-function relationship studies that would allow us to gain insight using biochemistry, gain insight about the structure of BCX1. And when you're talking about membrane proteins, oftentimes you think about how is it that these proteins, how is it that this primary sequence threads through the membrane, through biological membranes. And so one of the things that I did as a graduate student is that I determined using biochemistry and modifications at certain key sites along the protein, I determined its topology using different biochemical methods. And so I became an expert in biochemistry. I used yeast, Baker's yeast, as a model system within the context of biochemistry. And this was a very rewarding experience. So this was back in 2008, and as recently as 2013, I had the satisfaction of seeing the high-resolution structure of BCX1 being worked out by a lab, and the information that I had learned back in 2008 using biochemistry, it held true. It was supported by the high-resolution structure. So that was very rewarding. It really highlights this idea that when you are doing work that you enjoy, when you are tackling scientific questions that are interesting, even though time might pass, you still derive pleasure from seeing that field progress. And so then, as an older graduate student, as I was finalizing my graduate work, I became more interested in, well, great, I really like transmembrane proteins, but then the question was, how are these proteins getting to where they are supposed to function once they get made? And so I started reading up a lot of interesting work about membrane trafficking, this idea that the cell is equipped with pathways that transport materials, including membrane proteins, throughout the cell to where they're needed the most. And so that's where I developed, through self-study, a love for cell biology, even though I was trained as a biophysicist. And I decided to then look for opportunities as a postdoctoral researcher to nurture that love for cell biology and acquire experience in cell biology. And I decided to work to stay within the yeast world, especially because it's such a powerful system. As a graduate student, I learned how to handle yeast biochemically. I really wanted to explore how to manipulate genetically Baker's yeast. And so when I joined a lab for postdoc, those are the principles that guided my selecting of a postdoctoral mentor and lab. My postdoctoral research was very productive, and so my projects focused on the coat protein clathrin. Clathrin is a protein that helps transport vesicles be generated and helps transport of membrane proteins during endocytosis and also during protein sorting at the trans-Golgi or late Golgi network. So I did some work trying to identify new clathrin-coated vesicle cargo, and within that context, I came to better study and define a protein called ATG27. ATG stands for autophagy. And so I studied the biology of ATG27, its mailing addresses, so to speak, which parts of the protein had a say in where the protein was localized, and its contributions to the cellular process of autophagy. And that was a project that I started in my postdoctoral lab, and so I've had the opportunity. I've stayed within that niche now as a PI at a primarily undergraduate institution, or PUI. I had, of course, side projects having to do with creating different versions of clathrin that have properties that are different from wild type, that are useful to understand the mechanism of formation of transport vesicles. And I also did some work with oxalin. Oxalin is a protein that helps uncoat clathrin-coated vesicles. Once membrane transport vesicles are made with the help of clathrin, they have to be uncoated so that they can dock and fuse with the target membrane. And so oxalin helps do that. So very productive postdoc research, which guided then what my research lab is doing today. So these are a few pictures of members in my lab. That's my lab space, some of our students presenting at scientific conferences. And so I'll tell you a little bit more about what is it that we do and how is it that my lab uses the work and contributions of undergraduate students to advance our science. So autophagy is cellular self-eating. Is a set of processes that target materials for degradation. And that targeting can be very specific, like in mitophagy, or it can be in bulk, like in macroautophagy. And so in general, what you have going on during autophagy is that you have certain number of materials in the cytoplasm that you want to get to the degradative organelle of the cell for degradation. Okay, and so at a cellular location called the pre-autophagosomal structure, there is this transport vesicle specific to autophagy that is formed. Okay, it's seeded, expanded, and completed. This is then known as the autophagosome. So that now the autophagosome has sequestered the materials that you ultimately want to degrade. Then the autophagosome, once it's formed, it docks and fuses with the lysosomal vacuole in yeast, ensuing then the destruction or degradation of the delivered materials. And autophagy is a key process for surviving stress, stresses like starvation. So this is how cells would recycle components to sustain metabolism when they are experiencing stresses like starvation. Okay? There's a set of core machinery or ATG proteins that mediate the process. So now you might remember ATG27. And they all participate in this process. Okay? And so our lab studies autophagy in yeast. And this model system is great for autophagy because of it's easily grown, it's relatively cheap to grow, it's a very robust mechanism, and that's particularly useful for me because I am in an environment where undergraduates are the ones that are helping me drive my research forward. So the degree to which I can have them work on a robust, easy to handle system, I'm going to get further, and my results are going to be easier to get, less painful. Okay, so our main research focus is ATG27. So here we've zoomed in, so to speak, into the process of autophagosome formation. And so here's autophagosome, here it is as it's being formed. And we have an idea of how regular membrane transport vesicles contribute to autophagosome formation. I should mention that autophagosomes are very different than your canonical membrane transport vesicles. They're much larger. They are double membraned they're delimited by two different membranes, and so they're very, very characteristic of autophagy. And so during the biogenesis of autophagosomes, we think that transport vesicles budding off of the late Golgi contribute to the growth and provide membrane and protein for autophagosomes to form. And ATG27 is one of the proteins that helps during autophagosome formation. If you do not have ATG27, autophagy is less efficient and autophagosomes end up being smaller. So ATG27 is very important for the process. And it's very little. We know very little about ATG27 and what it's doing, okay? It's a very interesting protein. It actually, it sits on the membrane. It's a single pass transmembrane protein. It has a very small cytoplasmic domain and a very large luminal domain. We don't know what the luminal domain is doing. It must be doing something. It's conserved, but we don't know what it's doing. We know based on my work that I began as a postdoc that its cytoplasmic domain contains determinants for its localization, mailing addresses of sorts. And so now my lab is studying ATG27 function to better understand autophagy. Why autophagy has been associated with a variety of human diseases. When autophagy is not regulated properly, when it's not happening properly, it can lead to diseases like cancer, neurodegenerative disease as well. And so to the extent that we can understand the basic mechanism, we will be better able to understand how it can go wrong and how we can select therapeutic targets within the process to treat human disease. The process of autophagosome formation, it's one of the special processes about autophagy, and it's one of the least understood. And so since ATG27 is involved in that process, if we're able to understand what is it that it's doing, we'll have a better idea of what's going on. And so also during my postdoctoral work, we came to identify that ATG27 might have additional roles in cell biology as compared to just in autophagy. And so we're interested in looking to see whether there are any other alternative roles that are autophagy independent for ATG27 that have not been reported to date. And so here are, yes? Do you have a question?
Yes. Do you know what step of the cell cycle this must occur?
So it's during, so oftentimes the macroautophagy will be triggered or induced during starvation, stresses like starvation. So quiescent, when cells are quiescent. But there are more selective forms of autophagy, like mitophagy. I know that you mentioned that you study mitophagy to some extent. And more selective forms of autophagy target specific components within the cell. So mitophagy, if you could guess, is the autophagic degradation of mitochondria that are damaged. And so autophagy is also a great way for the cell to get rid of damaged components. So here are some of the undergraduate research projects that I have in my lab. So we want to map additional sorting signals on ATG27 that are present in that small cytoplasmic domain. Also ask the question, is the luminal domain of ATG27 involved in localization at all? That's an interesting question. Identify proteins that ATG27 might be helping get to the vacuole. As part of my postdoc, I discovered that when you have mutations in ATG27, that can lead to other proteins in autophagy being mislocalized, suggesting that ATG27 might be some sort of trafficking adapter for additional proteins. So can we identify additional protein cargoes for ATG27? In addition, that big luminal domain I mentioned, if you look at its sequence, its predicted three-dimensional fold, it actually resembles a mannose-6-phosphate receptor domain. Those are domains in mammalian cells that help sort specific types of proteins. And so the question is ATG27, does it have an MRH domain in its luminal region? And does that domain, is it required for autophagy? And so we have in the lab, undergraduates have not helped me generate mutants, point mutations that abrogate MRH domain function, okay, the predicted domain, and we are currently evaluating the mutants function in autophagy. Okay, and so it's been a great journey. So these are some of my lab alumni. So now I've been at High Point University, or HPU. This is going to be my third year. So I've trained quite a bit of students at the bench. And so here are some of our lab alumni. And I've also listed where they are now. So many of them have gone on to graduate school, not only at the bench in the sciences, but also alternative fields like medical illustration. And so, you know, as a research lab at a primarily undergraduate institution, we aim not only to drive our science forward, but also inspire students to use, to think like scientists and use those transferable skills that they acquire for whatever passion they have within their career and professional development. Okay, and so my lab is a little bit smaller now. Okay, so that's all about my science. So now you know about my science. How is it that I went from being a biophysicist, trained biophysicist, to a cell biologist? So let me tell you a little bit more about why I became, how did I become an educator? And I think that speaks to my creative side, okay? I wanted to create not only at the bench, but I also wanted to create in the classroom and in the community, in the context of science outreach. Why? As a graduate student, I explored professional aspects of being a scientist, and I really identified that I enjoy teaching when in small environments, when surrounded with colleagues that enjoy teaching as well, and when surrounded with students that want to learn. So I really like that interaction. I also experienced, while doing career exploration, I did some outreach work in the community, and I really like that. The idea that you can make contributions, concrete contributions, and increase the scientific literacy of your community. That was very, very appealing to me. So I became an educator because being an educator spoke to that creativity in me as a scientist. And then why a primarily undergraduate institution? I talked to you about my research, but the PUI environment not only allowed me to do research, but also, you know, allowed me to do the other things that I was passionate about. So teaching and science outreach. And the cool thing is that I get credit for it all. Okay, so there's not one thing that doesn't count, so to speak, right? Because we want to work hard, right? But we also want to progress as scientists and as professionals. So it was a strategic move, so to speak. Okay, so I want to tell you a little bit more about my teaching and about my science outreach. I get to innovate in the classroom. I get to create courses that had never been taught before. For example, last spring, a dream of mine came true. I was able to teach a 3,000 level cell biology course with a service learning component, with a science outreach component. So during this course, it was rigorous. It was 3,000 level biology, cell biology course. But during the laboratory component, students would create modules for science outreach at a nearby high school. And they got to create them, they got to test drive them in the lab, and they got to test them in the field, so to speak. What worked, what didn't. And in the process, undergraduate students get to discover what aspects of being a scientist they like. Do they like teaching? Do they like communicating with each other, with others? Do they have a hard time communicating? Is it something that they want to improve? And so all of these aspects of teaching for the real world, so to speak, help train, involve scientists. That is something that really speaks to me and that I have been able to do at a primarily undergraduate institution. Partly because our upper level courses are relatively small. So this class, you can see here my whole class for cell biology in the spring. So it's small enough that you can really experiment in the classroom as well. I also teach in the fall a general biology course for non-majors. So that one is not as small, but it's still very enjoyable. I get to meet students, okay, that are from all walks of life. They have all sorts of interests. Theater, arts, business, and I am challenged every fall to help them understand how is biology relevant to their life. Okay, and so I find that challenging, and I find that I enjoy the challenge. I enjoy the challenge. And so because I spend so much time teaching, okay, one of the ways in which I challenge myself is that as I am experimenting with new courses, new ways to teach, okay, I'm also doing scholarly work about my teaching. What worked, what didn't, okay? And so from there that I can publish on science pedagogy journals and let others know about the ways in which I am innovating in the classroom, okay? One of the areas that I really like experimenting with is using performance arts in the classroom. Concepts from performance art, for example, improv theater in the classroom. We had this yes and improvisational theater game that we applied in the classroom to help students talk science. Not only are they reading about new concepts and memorizing concepts, they're able to practice talking about these concepts in a fun way. Another aspect is science outreach, and this is an example of an outreach project that we did back last spring, so spring of last year, so 2016. Where our undergraduates collaborated with a nearby high school, Penn Griffin School of the Arts, to generate cell biology inspired art. And we had the students collaborate with each other over a period of 2 months, and the undergraduates helped high school students with their experiment, and they consulted with them as the high school students were generating their art. And so that was very, very rewarding. And then we concluded the long-term program with an art exhibit where the whole HighPoint community was invited, including parents. And so this is just a snapshot of what was going on in the room as we were presenting the different works of art that were generated by the high school students. To our surprise, not only did the students generate visual art pieces, but also music and dances and movements that highlighted some of the things that they learned about through experimentation. And so you can see, I love this picture because you can really see people that are picked with interest in the back. You might see some of them, dancing to tunes that were played about science. And so to me, that's great. Undergraduates are part of this. They're seeing how is it that as scientists they can have an impact in their community. And so here are some of the examples of the visual arts that were generated. Really cool stuff that was inspired by art, and also lasting relationships. So here we have an image of one of our undergraduates interacting with high school students. And so these are interactions that were forged during the program that are still going and are very meaningful. And of course, We need to disseminate what we've done, and so we are able to publish the types of programs that we create in appropriate journals. So this particular publication was published in the Science Teacher, which is a publication that it's considered a practitioner's journal. It's a journal for high school teachers that helps them innovate in the classroom. And so how great that our undergraduates at HBU were able to partake in giving ideas potentially to high school science teachers. And so then there's the component of persistence, right? So I think throughout my becoming a cell biologist educator, I've had to be persistent. And I'm sure that all of you, because you're here, you display some sort of persistence every day. And I think that's a skill that you improve over time. And so persistence to me is related to the following. When you fail, how do you recover from that? How do you get back on